Alright, here we have the TRP Operon. The TRP Operon is a repressive negative regulatory gene, or Operon, which means this Operon is normally turned on by default, or is constantly running. Here we have the TRPR gene. This gene will create mRNA. This mRNA will code for an inhibitor, or I'm sorry, repressor. This repressor is currently inactive. Over here we have the promoter, which includes the Tata box and the operator, and here we have the genes. Right now RNA polymerase is going through RNA synthesis, creating mRNA, which creates enzymes that will be used in tryptophan synthesis. Now, after this tryptophan synthesis occurs, there will be a higher concentration of tryptophan. Tryptophan will then bind to the inactive repressor, creating an active repressor. This active repressor will then bind to the operator. Therefore, RNA synthesis will cease because the RNA polymerase will not be able to go through and create mRNA. Alright, right here we have the LAC operon. The LAC operon is inducible negative regulatory operon. What this means is the operon is normally turned off. For example, this isn't used by bacteria such as E. coli. Generally, glucose is used for energy production. However, when glucose is not present, it will resort to use lactose as energy to produce energy. However, this lactose will not be able to be metabolized. Therefore, the LAC operon must be turned on in order to produce an enzyme that will be able to metabolize the lactose. Alright, right here we have the LAC inhibitor gene. This will create an mRNA, which will create an active inhibitor. This inhibitor is already bound to the operator, which is found in the promoter, right next to the Tata box. Therefore, RNA synthesis cannot be produced. However, when the lactose is present, allolactose will bind to this inhibitor. This inhibitor will misshape it and will not be able to bind to the operator anymore. Now, this RNA polymerase will go through and create mRNA. This mRNA will produce enzymes that will be able to metabolize the lactose. Alright, moving on to the CRP operon. As opposed to the LAC operon and the TRP operon, which were negative regulatory operons, the CRP operon is an example of a positive regulatory operon. Now, generally glucose is used for energy production in cells, right? So when this happens, there are low levels of cyclic AMP. However, when the cell has to resort to using lactose, the levels of cyclic AMP will be high. Now, when this happens, the cyclic AMP will bind with the inactive CRP, creating an active CRP. This active CRP will then bind to the CRP binding site, which is in the promoter, right next to the Tata box and the operator. Now, when this occurs, the DNA will slightly bend, and then it'll be easier to transcribe the DNA. Once that happens, the RNA polymerase runs through creating mRNA, and then this mRNA will create enzymes that will help break down lactose. Now, this is important because the CRP is also aiding the LAC operon, which does almost the same thing by breaking down the lactose. So, both put together would make an excellent combination into breaking down the lactose that is built up in the cell. Alright, here we have our RNA splicing. As opposed to the other three operons, such as the TRP operon, the LAC operon, and the CRP operon, the RNA splicing is only used in eukaryotes, when the other three are used in prokaryotes. Now, what we have here is our pre-mRNA that was taken from transcription of the DNA. And right here we have our complex, and then we have our final product. Now, what happens is the snRNPs are already present in the nucleus, combined with other proteins to create the spliceosome complex. What the spliceosome complex does is it attaches to the introns of the pre-mRNA, it excises them out, and then the exons will come together. So what happens is with the pre-mRNA has a 5' in the polytail, the exons and the introns. What the exons are, they are the expressed genes that is important, and that is what the product will have. The introns, however, are not very important, and therefore it can be knocked out. So then after it goes through the spliceosome, it will come out with the exons put together with the 5' in the poly A tail. And then the excised introns and the leftover components from the spliceosome complex will be able to be recycled and used over and over again.